Hi everyone and welcome back. Dear students, today we will start with the anatomy of individual plant organs separately for both dicots and monocots to explain some of the important differences in the anatomies of dicots and monocots. Today our focus is anatomy of dicot stem. If you will take a transverse section of dicot stem and place it under the microscope, you will observe the following structures. The first one is epidermis. It is the outermost protective layer which is made up of barrel-shaped cells. These cells are covered on their outer side by cuticle, and at some places it also bears multicellular uniserrate trichomes. Then the next region is hypodermis. Hypodermis lies below the epidermis and bears a few layers of collenchyma cells. In some cases you will find the hypodermis is constituted of two layers. The first one is collenchyma and the second one is chlorenchyma. Okay, you will see it in the image at the end. The main function of collenchyma is it gives mechanical strength to the stem. Then the next region is cortex. It is made up of it is multi-layered structure which is made up of rounded thin-walled parenchymatous cells with large or conspicuous intercellular spaces between the cells. Then the inner region which lies inner to cortex is endodermis. It is a single layer of barrel-shaped cells which are parenchymatous, and this layer separates the vascular tissues from the cortex. In case of stems, this is specifically called as starch sheath because these cells store starch in them. Then inner to endodermis, there is one more region which is known as pericycle. It is present on the inner side of endodermis and above the phloem of the vascular bundles in the form of semilunar patches. And these cells are made up of sclerenchyma. Semilunar patches means they are half moon shaped. Okay, you will see at the end. Then the medullary rays. In between the vascular bundles, there are a few layers of radially elongated parenchymatous cells. These radially elongated parenchymatous cells are known as medullary rays. Then the vascular bundles. The vascular bundles in case of dicot stems, they are arranged in the form of a ring, and each vascular bundle is conjoint, collateral, end-arc, and open. We have already discussed all these terms in our previous part. Okay. Then finally, pith. It is made up of a large number of rounded parenchymatous cells with large intercellular spaces. And this pith occupies the central portion of the stem. And the main function of pith is the storage of water and the food reserves. Here you can see in the image the outermost layer which is made up of barrel-shaped cells. It is known as epidermis, and these cells are covered on their outer side by cuticle, and at some places you will find a number of multicellular uniserrate trichomes. Okay, multicellular means they are composed of a number of cells which are arranged in a single row, uniserrate. Okay. Then inner to epidermis we have this collenchymatous hypodermis. Then one more region of hypodermis is chlorenchyma. Okay, these cells are chlorenchymatous, they possess chloroplasts. And inner to this collenchyma there is the general cortex. The individual cells of cortex, they are thin-walled, rounded, and they possess conspicuous intercellular spaces between them. Then the innermost layer of cortex is known as endodermis. The individual cells of endodermis, they are barrel-shaped and they possess Casparian strips on their radial and their transverse walls. We have already discussed all these things. Then there is one more region of cells which is known as pericycle. It is present in the form of semilunar patches. You can here see it is present in the form of semilunar patches, and the individual cells of this pericycle, they are sclerenchymatous. Then the medullary rays, these are the radially elongated parenchymatous cells. They are known as medullary rays. and then the vascular burners. The vascular burners, in case of dicot stems, they are arranged in the form of a ring. This is very much important. You have to remember this, that the vascular bundles in case of dicot stems, they are arranged in the form of a ring, and each vascular bundle is conjoint, collateral, and arc and open for secondary growth. We have discussed all these terms in our previous part, okay? And finally, in the center of the stem, you will find a group of cells which are large, they are thin-walled, they are parenchymatous, and this constitutes the pith of the dicot stem. This is all about the anatomy of dicot stems. In the next part, we are going to study the anatomy of monocot stems to understand the difference between dicot and monocot stems in terms of their anatomies. I hope you all understand this. Thank you and stay blessed.